Okay, I think we're at the top of the hour now, so I think we'll go ahead and get started. Again, welcome to everyone who's joining today's call. You are at a HIRN, the Human Islet Research Network webinar series. We've been doing these series every month about to try to promote the technologies and the informations and the approaches and a lot of the information that's being learned in HIRN to share with the community and get an opportunity to interact with other investigators, learn, hopefully make some collaborations. We have a fantastic talk today. You can see the title there. We have three wonderful speakers lined up, so I won't cover that. Let me just tell you a little bit about HIRN for those of you that are not accustomed to these webinars. HIRN was initially a network of 5 consortium, and the point of HIRN is to understand why beta cells are being lost in type 1 diabetes, and to try to understand how we can generate different approaches to different strategies in addressing the disease. NIH started this network, particularly NIDDK, back in 2014. And since then, there have been lots of changes because it was designed as a modular network. So people can come, people can go. And there were 5 different networks that were, or 5 different consortia that were initially created. Now, there have been some changes. There's a new initiative that has just joined HIRN, which is the Human Pancreas Knowledge Base. It's a very large initiative to bring together lots of data. So that's underway now. This isn't a network that's closed. There is an opportunity now in an RFA on the street open now. The RFA is out, and you can apply now. It's due in July, so there's still plenty of time to join. But over the years, there's been 800 researchers, 109 institutions, 12 countries. So you can get the idea that it's a pretty big investment. There are lots of funding opportunities, and if you're interested in learning about them, you can go to our website for all of these, where not only can you learn about what's happening at HIRN, but you can learn about the various job opportunities, labs in HIRN, people that have openings, whether they are postdoc positions or even faculty positions. Those are advertised as well. And I think I mentioned for you the latest initiative from HIRN that's on the street now, and you can see that, and you can always Google that. So also we have a website where there's not only information about HIRN, but we also have some resources available. And I just wanted to highlight a few of those resources again. Those are different funding resources that are on the upper right-hand side. You can see the funding there. You can go there. But we also have a number of other resources where we have databases. You can get lots of data on human islets. If you're interested in it, you can learn about the different antibodies that people are using, or different constructs that people have used through the years, different cell lines and such. And again, those are all available on the website as well. So those are things that you can access. And of course, you can follow us on the various social media platforms as well. Now, this is a resource browser. This catalogs close to about 3,000 different resources. You can see that the categories that they fall into. And so you can go and link through that through the website as well. You don't have to worry about the webinar. It's going to be recorded. So if you want to view it, we have a YouTube page where you can check it out. And see that, along with the many other webinars. If you've missed any of them, you can go back and you can check those things out as well too. So so just a few reminders again before we get started. I'm pretty sure everyone's already on mute, but if you're not, make sure that you're muted. We want to try to hold the hold any questions until the end. We should have plenty of time for questions. We've got a really nice agenda packed for you today. And so I'm just going to go ahead and switch off the screen. There seems to be a delay here, but while I do that, I'm going to hand off to you, and we'll switch screens here. So I'm going to introduce.
Hello, I think we lost John. So Saumya, would you like to get started?
Yeah, having a little bit of trouble sharing my screen here for some reason. Let's see if this works. Okay, here we go. Okay, can everybody see my screen and hear me?
Yes.
Great, okay. So thanks again for the opportunity to present some of our work here. We are relatively new to HIRN. We are mostly a cardiac, cardiovascular and metabolic disease research laboratory with a big focus on extracellular vesicles. But we recently got quite involved with the HIRN network through collaborations with Dr. Rohit Kulkarni, who's been a longstanding member, and have started looking into some of the aspects of the research we do as it applies to beta cells and beta cell stress. These are some of my disclosures, but none of these are really relevant for the presentation I'm giving today. So I thought I'd give a little bit of an outline of what I'll talk about. This is probably a topic that is not talked about a lot, or an area of great expertise for people in the HIRN network, although I'm sure some of you work with these extracellular vesicles. So I thought I'd give a little background on what these extracellular particles are, and in particular their RNA cargo, which we're quite interested in. And then I'll talk a little bit about the work we've already done, mostly in the cardiovascular field, in understanding the potential of these vesicles, really as sort of liquid biopsy approaches, as potential windows into disease pathways and disease transcriptomes in their cell of origin. Then I'll turn it over to Lingfei Sun, who is a research assistant in our laboratory for the last 2 years and has really done the bulk of the work in trying to apply what we have learned in cardiovascular disease to the specific case of beta cell stress. And she'll walk you through some of the methods we have developed and some of the initial findings we have. And then as the last speaker, Dr. Lee, who was a postdoc in my lab and is now running his own lab in our next to our group, has really focused on this novel species of RNAs called tRNA fragments, or tRNA-derived small RNAs, that appear to have an emerging role in beta cell maturation. But Guoping is going to talk a lot about how these might also are also present in the extracellular vesicles and are emerging as pretty novel markers of stress in different conditions. So that's sort of the agenda for today. Okay, so a little bit of background. Pretty much every cell type that has been looked at secrete particles. These are either extracellular vesicles that are delimited by membranes, but they can also include amembranous particles, and these have recently come into focus, mostly in the cancer biology field, that contain a variety of cargo. These vesicles can range in a large distribution of sizes. They may have different biophysical properties such as density, and their biogenesis, although not fully understood, likely are distinct. As I mentioned, the cargo of these vesicles and particles can contain many different moieties, but notably, we are particularly interested in small and long RNAs that are contained within these vesicles or bound to these extracellular particles. They may, of course, contain proteins that are unique and may reflect the proteome of the cell of origin, as well as lipids. And notably, in our case, where we are quite interested in using these RNAs that are present within these vesicles as biomarkers, they're fairly well protected from the circulating RNases in biofluids such as plasma. And more, there is emerging role for functional role for these extracellular vesicles and particles in mediating signaling both locally and across distance, across cell types and organs. These are just some of the papers. The diagram below actually shows that this is not a completely new field. So these are reticulocytes, And back in 1970, people showed that they shed non-functional or proteins that are no longer needed, such as ferritin, as they mature from reticulocytes to red blood cells. And then this figure on the right hand is a paper that was one of the first papers that showed that fluorescently marked vesicles can track from cancer cells and mediate distal metastases. So sort of a whole journey back from 1970 to now, which has really evolved the field. So as I mentioned, there's a whole wide diversity of these particles, and this graphical representation on the left just gives you an idea of how these can go from really large vesicles, including ones that are secreted from cancer called oncosomes, to slightly smaller microvesicles, and go all the way down to these really tiny particles called supermeres and exomeres that have only really in the last 2 or 3 years come into existence or been discovered as being entities that are likely important in many types of disease pathogenesis. And in the middle is the ones that we mostly focus on, called small extracellular vesicles that are in the order of 60 to 120 nanometers and have pretty canonical markers on their surface that allows us to identify them as such. As I mentioned, their contents can be quite diverse, but include small RNAs such as microRNAs, as we learn about from Guoping, tRNA fragments, but also proteins, mRNA fragments, and even DNAs. This paper sort of showed that, in fact, if you look across these different particles, they can be marked by different types of proteins. And this was a proteomic study that was done from a cancer cell line that isolated quite rigorously all these different particles through a rather tedious method that takes 2 to 3 days of repeated ultracentrifugations, and showed that if you look at what's contained in these, that there are pretty distinct signature of proteins that are in these small little particles called exomeres and supermeres versus small extracellular vesicles and larger vesicles. And that's sort of shown here as a prototypical marker. You can see that there are proteins called such as TGF-beta I that's really heavily represented in supermeres. Syntenin, which is a classic marker for extracellular vesicles that are present in, as noted, in extracellular vesicles, and things like flotilin and HSPA13 that again have distinct sort of signatures. And these really have some clinical relevance. So here is a follow-up study that these same authors did, where they followed patients with a high level of supermeres versus low level of supermeres. These are cancer patients, and showed that there was a clear difference in survival in patients who had different levels of these supermeres as defined by these particular markers. And just like they have different protein levels, these different types of particles have quite distinct signatures for small RNAs as well. So for example, microRNAs and RNPs are highly enriched in exomeres and supermeres, and that's sort of shown here, where you see microRNAs in blue that are really enriched in exomeres. It's doing that. And then you have things like tRNAs that are much more expressed in small EVs. And you have snow RNAs that are quite expressed in supermeres. So fairly distinct and unique signatures for these different types of RNAs. And if you look at individual microRNAs, for example, you can also find defined signatures for supermeres versus exomeres versus these small EVs. Of course, the EV microRNAs were the ones that were most studied initially because they were much easier to assay. We knew a lot more about microRNAs than we do about these other small RNA species. And we spent a good amount of time really understanding if plasma microRNAs or microRNAs contained within vesicles can be related to clinical phenotypes that we were interested in. And these have been published in multiple journals, looking primarily at cardiac phenotypes, so patients after myocardial infarction, how these microRNAs present within vesicles could be correlated to phenotypes such as sudden cardiac death, or the size of the ventricle, or how the ventricle remodels or functions. And then related to insulin resistance and metabolic phenotypes, we had also published that, for example, a microRNA that comes from the liver called microRNA 122 is quite well correlated with quartiles of insulin in patients across the spectrum of metabolic diseases. So we had fairly good evidence that these plasma microRNAs could be used as biomarkers in certain cases to define phenotypes of interest. The question then becomes, how come after 15 years of studying these, have these not really become novel biomarkers for disease that have been translated to the clinical arena? And part of the issues are that there have been technical variability of assays when these studies were initially done that have steadily been improving. But really, one of the biggest issues is that small RNAs, such as microRNAs, have a rather promiscuous tissue expression. So it's hard to define them as specifically reflecting disease pathogenesis in particular cell types where these EVs are coming from, because they're expressed in many other cell types as well. And so therein becomes the problem, because if you look in biofluids such as plasma, most of your vesicles are going to come from hematopoietic cells, a good degree from the liver, but perhaps a small amount from tissues that you're interested in our case, for example, heart cells or beta cells. And so when you're sampling just in plasma EVs or plasma RNAs, your signal that you're really interested in coming from the cell type of your interest may be drowned out by noise that is coming from all these other cell types. And so the overall effect size might be quite small when you do that. And so we set about trying to figure out how we could improve a representation of the stress in the cell type that we were interested in. And herein we had a study where we switched from small RNAs to mRNAs that can also be present within these vesicles. And we found that, in fact, mRNAs, much better than small RNAs, can represent the transcriptome of the cell much more accurately. And so these are correlations in HEK cells at baseline are subjected to hypoxia. And you can see that the correlation is quite good. And if you look at RNAs that change with hypoxia in particular, these are all downregulated and these are upregulated. You can see that the correlation is really quite excellent between what these RNAs do within the cell and how they are represented in EVs itself. And so based on this information, we switched a little bit to looking at what happens to RNA or the transcript of EVs in heart failure. And so heart failure is a condition we are quite interested in. There are two different subtypes that are actually quite different. Heart failure with reduced ejection fraction is primarily a disease of the heart, and heart failure with preserved ejection fraction, where the heart overall squeeze of the heart is not affected, is really a systemic disease with patients who have metabolic syndrome, hypertension, et cetera. And so we expected that perhaps these two will be quite different if our theory held true. And in fact, we can tell that there are many different RNAs that are different in patients with heart failure with preserved ejection fraction compared to control patients, and that these are not the same as the ones that are different in heart failure with reduced ejection fraction, which is, again, a heart-specific disorder compared to control patients. And this is represented when you try and map these RNAs. So if you ask, where are these RNAs coming from that are in these vesicles that are circulating in the plasma, you see that in heart failure with reduced ejection fraction, most of these RNAs that are present that are different between heart failure and control patients come from the heart. Whereas when you look at this more systemic disease of heart failure with preserved ejection fraction, they appear to come from many different organs, including adipose tissues, inflammatory cells, kidney transcripts. And then you can ask, like, if you map this to single nuclear sequencing data, I'll go into this a little bit, from patients. So this is data that is derived from heart tissue from patients with heart failure with reduced ejection fraction. Most of the transcripts that are changed in EVs really map to cardiomyocytes. And this sort of makes sense biologically because that's where most of the action happens in heart failure with reduced ejection fraction. If you now map these transcripts with heart failure with preserved ejection fraction, however, the situation is somewhat different. And you can see that these transcripts map, certainly some of them map to cardiomyocytes, that's shown here, but a lot of them map to many other cell types, including adipocytes, immune cells, macrophages, and endothelial cells. So this suggested that when you start sampling these mRNAs, unlike small RNAs that are much more promiscuous in expression, you can begin to get a better idea of where these are coming from and how they might change with different diseases. And this is just an example of how we did this mapping. This is the single nuclear atlas from, again, patients with a low ejection fraction or HFrEF. These are patients with a dilated cardiomyopathy. This purple blob you see are cardiomyocytes. And you can see most of these are mapping into cardiomyocytes, like I showed you with the upset plot in the previous slide. When you look at HFpEF patients, you see that not only, certainly some of them map to cardiomyocytes, but a lot of them map to other cell types, including, for example, endothelial cells, immune cells here, adipocytes, fibroblasts, etc. So this sort of really reflected what we know about the disease, but really gave us an idea that these EVs might really represent liquid biopsies. And these could be quite clinically meaningful. So you can see that we can get excellent discrimination using these RNAs that are present within vesicles between HFpEF in control or HFrEF in control. And these really are quite good for the field in terms of AUC for discrimination. So as I mentioned, one of the other issues in the field has been that the majority of circulating vesicles are going to come from hematopoietic cells and other organs that we're not so interested in, particularly if we're interested in, for example, the pancreas or the heart. And so the last step we did was to say, well, is there a way to isolate specifically vesicles that come from the heart? And to do this, we did a proteomic approach where we isolated vesicles from iPSC-derived cardiomyocytes, isolated them so they were quite clean, and then there's mass spec on them to identify proteins that might be on these vesicles that are specific to the heart. And when we do that, we found actually 2 proteins that are present in cardiomyocytes, highly enriched in vesicles. And if you map them again to this single nuclear atlas, we find that they're quite expressed in the heart, or in the case of cranie, to the heart, as well as some other heart cell types. But when you map them across the body, again, these are quite enriched in the heart, although again, there may be some expression in other cell types. So we will call them cardiac enriched proteins that are present in vesicles. So we then said, well, if you isolate these vesicles from patients, and we, over a couple of years, devised this rather complicated method, but one that really results in fairly pure vesicles that contain these EVs. These are examples of pull downs that we're able to do immunomagnetic separation using antibodies against POP-TC2 or CRINE. These are the 2 antigens on these EVs that come from the heart. And then we sequence these EVs, and we find that most of the RNAs that are within this, anything to up on this side of the line, is enriched in heart cells, really are quite enriched in heart cells. And a few examples are shown here where you have transcripts that are present in the 2 different types of vesicles or control EVs. And you can see that they're quite enriched when you look at counts per million in heart cells. So the last step was to say, well, how does that change with disease? And so we went back to patients with 2 different types of diseases, and I'll only show you one for the interest of time. But here, again, these are isolated from control patients here. And you can see that when you isolate from the control patients and map it to the entire body single nuclear atlas, it lights up in the heart. So the blue part is the heart. So these transcripts are really quite specific for the heart. And then within the heart, again, you can see it lighting up in cardiomyocytes, although with one of them, it lights up in a few other cells as well. So the last slide I'll show is that, well, what happens with disease? So here is myocardial infarction, ischemia to the heart. When we isolate the vesicles from this and ask what pathways are enriched in these RNAs, the top hit in both 2 different ways of analyzing this is myocardial ischemia. And this is kind of shown in some of the proteins that are changed. I'll say that a large number of them overlap between these 2 types of vesicles, because some of these are present in both vesicles, but they're overwhelmingly enriched in pathways of myocardial ischemia, suggesting that if now we are able to isolate vesicles specifically coming from the heart in these patients, we can detect pathways that we think are important in biology, suggesting that this could be a liquid biopsy sort of pathway. So I'll turn it over to Lingfei now to see how we've applied this to type 1 diabetes. So Lingfei, you may want to unmute, and I'll drive your slides if you just tell me.
Thank you so much for unmuting me, and thank you, Dr. Das, for giving me a opportunity to talk. So I'm going to talk a little about the emerging role for tissue-specific extracellular vesicles in disease diagnosis, implication for T1D. All right, next slide, please. All right, type 1 diabetes is a chronic condition that the pancreas makes little or no insulin. It is caused by autoimmune reaction, meaning that the body attacks itself by mistake. This reaction destroys the beta cell in the pancreas, and this process can go on for months or years before any symptoms appear. Right, next slide, please. The development of type 1 diabetes occurs in 3 stages. Stage 1 is asymptomatic. In this stage, people who later will be diagnosed as T1D will have normal fasting glucose, normal glucose tolerance, and the presence of greater than or equal to 2 pancreatic autoantibodies. And stage 2 will include the presence of the greater than or equal to 2 pancreatic autoantibodies and with impaired fasting glucose or impaired glucose tolerance. However, at this stage, the individuals still remain asymptomatic. In stage 3, there's diabetes or hyperglycemia with clinical symptoms and 2 or more pancreatic autoantibodies. We focus on finding the biomarkers that can identify early stage 1 or even before stage 1, as this is the asymptomatic stage and most of the beta cells are still functional. In this case, there will be lots of strategies to prevent the autoimmune attacks and try to preserve this beta cell mass. Next slide, please. And our aim is to use human beta cell tissue to identify and to characterize cargoes of pancreatic beta cell specific extracellular vesicles. To do this, we have 4 steps. The first step is to identify beta cell EV membrane proteins, both from the NOC beta H1 cell lines and then the human islet EVs. And the second step would be to perform immunocapture, also known as pull down and RNA sequencing from EVs from NOC and islet with or without cytokine stress. And 2 would be for the control and type 1 diabetes patient plasma EV. And the third step is to identify potential beta cell EV RNA cargo for early diagnosis of T1D. And the last step would be to validate in a larger cohort and to study the function of the EV RNA identified. All right, next slide, please. So we first chose to use NOC beta H1 cell lines first because it's a robust and immortalized human beta cell line, which mimics the physiology of primary human cell. And they're very helpful for us because they express many beta cell specific markers, and it can be served as a unique tool for large-scale drug discovery and provide a preclinical model for cell replacement therapy. Since we have this cell line in hand, so we went ahead and cultured the cell line and collect the conditional media for EV isolation. Next. All right. And there's due to the concurrent presence of the lipoproteins, ribonuclear proteins, and other nanoparticles, EV isolations can be challenging. So the lipoproteins are abundant in the bloodstream. There are around 20 to 100-fold more lipoproteins than EVs in plasma isolate. And there are 2 different methods to isolate EVs. The first method would be separate EVs by density. However, as shown by the graph, if we do this method, the EVs will be co-isolated with HDL. This is high density lipoprotein. And the second isolation will be isolated due to size. However, the EV will be co-isolated with VLDL, very low density lipoprotein. So therefore, we will use a 2-step method. Can you go to the next slide, please? Thank you. We will use a 2-step method for EV isolation to ensure the EVs that we get are as pure as possible. So first, we utilize the cushion density gradient ultracentrifugation method. This method allows us to remove the lipoprotein by density, and then we will continue with size exclusion chromatography using the eyes on QEV column. This method allows us to remove the lipoprotein by size, so we can remove the HDL. And even though the cell conditional media from NOC beta H1 cells don't have lipoproteins, we still use this 2-step method to make sure we're isolating EVs the same way as how we will isolate EVs from patient plasma sample later on. Next slide, please. All right, and those are the EVs isolated from the NOC beta H1 cell conditional media. Here's the MRPS graph from SpectraDyne which tells us that there are around 2 times 10 to the 9th particles per ml of the conditional media. We observe most particles around 65 nanometer, which is what we have expected. And the Western blot here confirm EV identity, since you can see all the cargo proteins, ALEX, TSG101, and syntanine. All right, next slide, please. And after we have confirmed the EV identity, we send out for proteomic studies. So first, we identify 1,262 proteins in total, and there are 466 proteins are expressed in pancreas, in which 203 of them are membrane proteins. Since we want to do immunocapture later on, so we need to have the membrane proteins. And further analysis overlapping these proteins with human protein atlas indicates that there are 13 proteins potentially to be pancreas specific, of which 5 of them are membrane proteins. So for confidentiality reasons, we're not going to disclose the name here, so we're going to be talking as protein A to E. And thus we order a bunch of antibodies to see if the identified 5 membrane proteins are beta cell specific. We weren't able to find any antibodies for protein B, so we skipped that. For the pilot testing, we first use hex cell lysis as the negative control. Protein A, C, and E appear to be specific to beta cell, while protein B isn't. This only is a pilot testing and working with Dr. Kokomri to get more human ductal and arsenal cell lysis to confirm the beta cell specificity of these proteins. Next slide, please. And then we want to confirm the select membrane protein is on the EVs derived from the NOC beta H1 cell. And then we run a Western blot. As we can see here, the protein A, C, and E are expressed here. And the insulin shows that there's no cellular contamination. And the cargo protein, ALEX, syntanine, and the tetraspanin protein, CD9, confirm their EV identity. And now we want to do the same thing on the human islet EV. So we culture the human islet EV for a week and collect the conditional media every 24 hours. We spun out the cell debris and exclude the particles larger than 0.8 micron. The same methodology is used to isolate EVs. And here's the MRPS graph from SpectraDyne. This time tells us we have around 5.8 times 10 to the 10th power particles per ml of the conditional media. We also observe the most particles around 65 nanometer, which is consistent. And the Western blot here confirm EV identity, since we can see the cargo proteins, ALEX, syntanine, TSG101, and also the 2 tetraspanin proteins, CD9 and CD81. GM130 is a Golgi protein, and this ensures us that the islet EVs don't have any cellular protein contaminations. All right, and then we send out for proteomic studies again. And this time we identify 1,026 proteins in total, and 844 proteins are expressed in pancreas, in which 315 of them are membrane proteins. We also did a further analysis overlapping these proteins with human protein atlas, indicate that there are 17 proteins potentially to be pancreas specific, of which 3 of them are membrane protein. And 2 of the proteins, protein A and C, were also identified in the previous NOC beta H1E EV studies. So this is very good news to us. And afterwards, we want to confirm the select membrane proteins is on the EV derived from human islets. So we ran a Western blot again. From here, the CD81 and ALEX confirm the EV identity. We can also see the protein A expressed on the islet EVs. Right, now we want to perform immunocapture to see if we can pull down the EVs from both the NOC beta H1 cell conditional media and the human islet EV using the protein A antibodies. Based on the Western blot, we saw that we can successfully pull it down. Also, CD81 ALEX confirmed the EV identity. So that's good news. And the future direction would be to pull down EVs derived from plasma and serum and send them out for RNA sequencing to see if there's any potential beta cell EV RNA cargos for early diagnosis of type 1 diabetes. And after that, we'll validate in larger cohorts and study the function of the EV RNA identified. And at this time, I'm going to give the floor to Guoping for more RNA talk.
Okay. Could you see my screen?
Yes.
Okay, great. So it's good to talk about my work regarding the novel extracellular biomarkers. At this time, it appears that we can perfectly isolate the tissue-specific EVs from the plasma samples, especially like in this case, we're able to isolate beta cell-specific EVs, and we want to characterize what's the potential biomarker that could be used to predict the earlier diagnosis of type 1 diabetes. In addition to the biomarker, the RNA species that Saumya just mentioned, like microRNA, mRNA, and long non-coding RNAs, we devote a lot of time to explore new RNA species that can be a better candidates for biomarker study. This is the multiple center effort that we published a couple years ago, that we sequenced the different human biofluid and together with conditional cell culture media to explore that potential other novel RNA species. In this case, aside from microRNA that can be detected, we found that a big significant portion of novel RNAs called tRNAs. It's the purple bar here. And when we look into these sequences, it turned out to be these are tRNA-derived small RNAs, we call dsRNAs or TDRs. Like in 2000, starting from 2008, a lot of emerging investigators has found that these tRNAs could be cleaved by several ribonucleases to generate smaller fragments. And these fragments are not simple tRNA debris, they are functional small regulator RNAs. It has been reported to involved in diverse biological processes like the regulation of transcription, translation. Few of them are included in stress granule assembly and apoptosis. An earlier study just published in 2022 reported that these 5' TDRs are responsible for the maturation of beta cells. So I want to point out that characterizing the tRNA is not a simple work. It is very challenging technically, because the presence of modifications on tRNA, especially the methylation, usually will cause the reverse transcriptase. Because during the cDNA library prep, that means with the conventional small RNA-seq, you can rarely or only able to capture a small subset of the tRNA from any cell total RNAs. In 2050, there's a group from our collaborator, Dr. Louis from UCSC, developed this optimized tRNA sequencing called RNAseq, which leverages the pretreatment of RBKB to remove the methylation and facilitate the RNA sequencing. So we have established this workflow in our lab and tested out with a few samples. The result turns out to be very good in capture TDR rates. This is the full-length tRNA. So as you can see with the conventional small RNA-seq, usually most of the sequence reads stop at position 58, which is a well-known M1A modification here. But with RNAseq, we can able to detect the sequence reads across this line and get more diverse and deep information about TDR rates. So after confirming this workflow in our lab, we designed this study that we want to know how these extracellular TDR are regulated upon different stressors, and whether it could be a novel biomarker for our patient biomarker exploration study. So we choose 4 common used cell type in our lab and expose them to 3 common stressors, including nutritional deprivation, hypoxia, and oxidative stress. Then we'll collect the cellular RNA and extracellular RNA and subject them to RNAseq. The sequence reads were then analyzed by T-Rex pipeline that was developed by Dr. Louis. And when we check some basic characteristics about the TDRs, including both cellular and extracellular, it turns out a very interesting phenotype here. As you can see here, the blue dots, the blue line includes the cellular tRNAs, and the extracellular tRNAs is labeled as the orange line. Interestingly, across every cell types we examined, every stress conditions, most of these extracellular TDRs has a very unique length. It's about 31 to 33 nucleotides in length. And when we map to the tRNA positions, it appears to be tRNA halves, with 70% of them derived from the 5' end and 30% derived from the 3' end. Interestingly, in the 3' end, it has a specific earlier end termination. So when we analyze the end position of the TDRs, consistently the 5' ends an anticodon loop at position 31 to 33, and the 3' TDR ends at a position 74, while the intracellular TDR ends usually at 76. So now, next we want to know like how dynamic is this TDR regulator upon different stressors in both inside the cells and outside the cells. This is the dots that indicate each TDRs. We track them using a line. As you can see here, there's a significant amount of TDR that are regulated upon stress, both inside the cells and out in the EVs. It's much dynamic in the extracellular area samples. So also it's consistent here, as you can see, the extracellular TDRs are usually not expressed in the cells. It's consistent with our previous finding that extracellular RNA TDRs is unique. It's but if I put this one here, this is the microRNA expression. As you can see here, it's not that significant or dramatically regulated upon stresses. So to further confirm this, we have done this PCA blood clustering and see whether the extracellular TDR signature could be better than the microRNAs to distinguish these stressors. So this is the simple PCA plot. As you can see here, the different color indicate different stressors, and the triangle means the extracellular samples. So this is the PCA plot based on the TDR expression profile. You can see here, these stressors are well separated using TDR signatures. However, the microRNA signature can't separate them very well, even though they could separate the cellular samples. It's same in the other cell types, the cardiomyocyte, the cardiofibroblast, in the vivo cell types. And it's also consistent in using different algorithms like UMAP or t-SNE or Spearman correlation assay. So this turns out to be that extracellular TDRs could provide better discrimination between different stress response compared to the microRNAs. So it means when you're using extracellular RNA samples, the TDR expression may be a better candidate than extracellular microRNAs. So we merge all the cell type together, separate them to human and rat because they are different species. And this is the comprehensive stress-specific extracellular TDR signature we built. And we have 3 different nutritional deprivation specific, hypoxia specific, and oxidative stress specific. I will not go deeper, but we are urgently want to explore whether this is true in the human patients. So we choose a patient sample that the patient's undergoing cardiac surgery, and it has to be connected to the cardiopulmonary bypass machine. So it is well known that when the patient is connected to the cardiopulmonary bypass machine, it will have an ischemia condition inside the human body. So we collect the samples from the same patient at 2 time points. The first one was collected before connected to this CPP machine. And about 70 minutes after, when we collect the sample immediately before the disconnection. So we sequence both the samples using RNAseq. This is what we got. As you can see, we can detect about 10% of the sequence reads are aligned to tRNAs. As you will imagine that we haven't done a ribosomal RNA depletion with so still a decent amount, like 50% of them are aligned to ribosomal RNA. So if we can deplete those, the tRNA reads will significantly amplified. We also check the basic tRNA profiles in these human plasma samples. It's consistent with our in vitro data. Most of the plasma TDRs range at 31 to 35 nucleotides in length, and it's also tRNA halves with 5' halves and 3' halves. And the end position is still in the anticodon loop with the 5' TDRs and at 74 with 3' TDRs. Interestingly, it has also have a unique population here. It's around 16 to 18 nucleotides in length that we don't know what they are yet. So, but overall, it's very consistent with our in vitro study that the extracellular TDRs has a unique fragmentation pattern. So we want to know whether it could be a better biomarker. This is what we did similar at the intracellular in vitro data. So we use the PCA plot to track the changes before connected to CPP and after connected to the CPP. So the red dot is before the connection and the blue dot is post the connection. As you can see here, using TDR expression profile, these patient could be well separated before CBP connection and post CBP connection. But using microRNA is kind of overlapped. It's not easy to separate them very well. In addition to this separation, we also try to see whether it could map to our the stress-specific extracellular TDR signature that we built using our in vitro model. So we have detected about 110 differentially expressed TDRs before during the connection to the cardiopulmonary bypass machine. And when we map to the TDR atlas we built in the human cell culture model, interestingly, these 110 differentially expressed TDRs cannot overlap with the hypoxia, but it has a well overlap with the oxidative stress and nutritional deprivation. So this indicates that when the patient connected to the cardiopulmonary bypass machine, it has the stress response related to the oxidative stress and to nutritional deficiency, but not hypoxia. And so when we go back to the surgery itself, it is quite reasonable because when we transfuse the blood back, usually we'll give the blood oxygenation. So that's why it doesn't have a hypoxia response in the human body. It turns out that this is just some examples showing that individual TDR that are significantly changed, even with the 70 minutes connection to the cardiopulmonary bypass machine, it is already changed. Like this one, tRNA 38 to 74 position from origin TCT can only be detected when connected to the cardiopulmonary bypass machine. So this, I want to summarize here that I hope you can appreciate that we demonstrate the RNAseq works better than conventional RNAseq on detecting TDRs. And extracellular TDR has a very unique fragmentation pattern when compared to the intracellular TDRs. And importantly, the extracellular TDR landscapes could provide better discrimination between different stress response compared to the extracellular microRNAs. And the stress-specific TDR signatures could be used to predict the stress response in the human body. That means these extracellular TDR could be a novel RNA species that could be used to emerge as a novel biomarker to predict different diseases, including the type 1 diabetes. So that's majorly our next steps to sequencing the beta cell specific EVs using RNAseq to try to see whether these extracellular TDRs could be a promising biomarker. So this is our acknowledgement. Our thanks the whole lab and the collaborator like Rohit, Patrick for the proteomics, and totally for all the bioinformatics support and the funding agency. Thank you.
Thank you very much, Guoping, for that presentation, Lingfei for your work as well, and Saumya for giving us that wonderful overview of everything going on in your lab. If all three of you would stay on camera, and I'd ask the audience, if you have questions, can you go ahead and raise your hand? You should have the ability now to unmute yourself, but raising your hand helps to stage the question so that we can kind of have an order to those. So while we wait for questions, great, Farouk, I see yours. Lingfei, let me ask you one. When you were showing your elevated EVs, I'm sorry, the elevated membrane proteins in the endo-Cs, and then in the islets, you had 5 unnamed proteins on the one hand, and then 3, and then you went into that protein A that you were looking at. What happened to, I think, what you call protein C and protein F? And is the conclusion there aren't that many membrane specific proteins that you can use to mark these EVs? I mean, even though you've shown a lot of elevated like transcript levels there, but at the end it sort of boiled down to a really finite, and I guess thankfully a handful of them. But what's the conclusion from that?
Since that we don't have as much as the membrane proteins, and then even though sometimes we have the membrane proteins, there aren't a lot of available antibodies to detect those membrane proteins, and some of the antibodies doesn't recognize the extracellular domain of that protein, so it would be hard for them to use them for immunocapture because the antibody will have to recognize the extracellular part so that we can pull down the vesicles.
Okay, terrific.
Yeah, John, I'll add that there are a lot of other membrane proteins, but we were particularly focused on ones that were really enriched in beta cells.
Yes.
So obviously there are others that may have some degree of enrichment, but we used a pretty rigorous cutoff to really try and hone down onto beta cells, because we may have to liberalize it if it doesn't work particularly well in plasma, but initial indications are that we can in fact pull it down in plasma in normals even. So hopefully that'll work, and thankfully we don't have to try and screen through different proteins.
Right, that's the blessing on the one hand, right? Farouk, I see you have a question.
Yeah, thank you, John. It's amazing talk. Really, I enjoyed it. The question I have is, Saumya, for you about the RNA, right? mRNA. So do you see like full fragmented RNAs, or these are fragmented RNAs that are were digested through RNAs, and then you find particular length of RNAs?
Yeah,
is that
it is a great question. This is actually an area of great interest to you. You picked up on that. There are some full length smaller RNAs, link RNAs and such. The majority are fragments, but they're not random fragments. So they're not just products of RNA degradation. In fact, there are particular fragments that might be specific to a disease type. So one of the lessons we have learned from that is, by sequencing, you may detect something. You cannot just use a set of primers and try and validate that. You actually have to do what we call, we map the transcript to figure out which are the fragments and what the coverage is. So we generate coverage plot for every RNA that we're interested in. And doing that, we found that it's almost like, in the old days, we used to do nuclear footprinting experiment. I don't know, I'm dating myself, because with graduate school, we used to do those where we had to make radioactive RNA and then incubated with proteins. And so these almost look like that so I think it reflects probably binding of certain RNA binding proteins to these mRNAs that protects those fragments while the rest is degraded, and somehow maybe that's what's transported in. But they're really patterns of fragmentation that are very stereotypical for the disease you're looking at.
I see, because I was asking this question about Ron Kahn's paper on microRNA, right? Whether there is particular motifs which these RNAs have, so they can be, sequestered into the endosomal vesicle and then released into thing. Not all RNA might have the same kind of thing. Yeah, that's amazing thing. Could you able to explain whether you could able to see these things increased during type 1D onset? Have you tested in the clinical samples?
Yeah, that's the plan. So we sort of approached it systematically in a rigorous manner to make sure we don't get confounders. So I think what Lingfei now is doing is we have obviously some of these samples are really precious. We have the last phase of our grant is the samples from the TEDdy cohort that are young patients. Those are extremely precious. So Rohit has actually generated a biobank of much more plasma sample in patients around the time of seroconversion. So we are trying to, or just before seroconversion, but mostly around the time of seroconversion. So we're trying to isolate from that next to do the sequencing and compare them to age-matched patients who don't have type 1 diabetes and see, whether we can start to detect pathways that have been previously shown in other types of studies. But in particular, we want to try and map it the same way we did in our heart studies to see if we can map it to a single nuclear atlas of islets, for example, with or without stress.
Amazing. Thank you.
Terrific. Wonderful. Thank you, Farouk. I appreciate the question. Again, if you have any questions, please raise your hand or unmute yourself so I can see you in line effectively. I didn't get the chance to introduce you, Saumya, but I wanted to tell the audience you are a member of HIRN. As you mentioned, you do have a few grants on this. You have an R01 specific to this project, but you've also, as you mentioned with Rohit, you're looking at various models. You're part of the HPAC consortium as well with other investigators. So you're taking that cardiac expertise and all that data that you showed us in the beginning, and you're applying it here in type 1 diabetes, which is really nice. Thank you.
No, thanks for having us be sort of honorary members of HIRN, and we've learned a lot from the last meeting as well. Just really great biology.
Yeah, that's terrific. I don't see any more questions coming in. If there's any, let me just check the chat and make sure that nothing came in the chat there. Yeah, I don't see anything else there. So going once, if I don't see anything else, I just want to thank the three of you again for giving us this wonderful overview of this data, and especially going back to the history. And what I enjoyed was the limitations, why things haven't caught on, and you addressing that and explaining that's very helpful to me, and I'm sure to other people in the audience as well. So thank the three of you for giving this talk, and we look forward to an update soon. Thank you.
Thank you.
Okay.
Thank you so much for having us.
Yes. Thank you, guys.
Bye-bye.
Bye-bye.